It gives me enormous pleasure to welcome our next plenary, Nalini Puniamoorthy, Assistant Professor from the National University of Singapore. Nalini did both her undergraduate degree and her master's degree at the National University of Singapore, before completing her PhD at the Institute of Evolutionary Biology and Environmental Studies at the University of Zurich. She then went on to do postdoctoral research at the Center for Reproductive Evolution at, I can never pronounce this one, Syracuse University, probably got that wrong as well. Where she, I guess, kind of fine-tuned and has done some excellent work around the causes and consequences of pre- and post-copulatory sexual selection. So really, she's primarily interested in what drives the evolution of reproductive traits, and also what are the consequences of variation between populations in these trait evolutions in relation to reproductive isolation or reproductive barriers. Okay, so it gives me great pleasure to welcome Nalini.
So before I start, can I just check if the folk on Zoom can hear me? Can hear me? You could just give a thumbs up or raise hands. Okay, perfect. Thank you. So then without further ado, let me get started. I just wanted to say, oh, there's a chat. Oh, thank you, Tegan, who said they can hear me. Perfect. So first off, thank you for turning up after last night's events. So I appreciate your physical attendance. And also thanks to the organizers for inviting me. It's been a really great few days chatting with like-minded people who use the same vernacular. So I've been really excited. So what I'm going to be talking about today is the work that we are currently doing at the National University of Singapore, where I'm leading the research at the Reproductive Evolution Lab. And one of the driving questions is what drives biological diversity, right? So understanding the ecological processes and evolutionary forces that mediate phenotypic divergence across lineages is a long-standing interest in evolutionary biology and in biology in general. And there are many, many barriers that can arise that can mediate barriers, that can act as barriers to gene flow. Now, these barriers can be induced by the environment, actually physical separations or geographical barriers. There could be ecological barriers that arise, let's say, due to local adaptation or niche specialization, or even temporal isolation, seasonal differences. But there's also reproductive barriers that can arise in sexually reproducing organisms. For instance, there can be pre-copulatory barriers that arise due to, let's say, mating behavior, courtship, and there can be post-copulatory barriers that arise due to post-mating pre-zygotic mechanisms like sperm competition or zygote viability that comes from, let's say, Dopansky-Mueller incompatibilities or negative epistasis. So one of the processes that can be really strong in driving phenotypic divergence is sexual selection, which basically refers to differential reproductive success that leads to differential fitness based on both mate acquisition as well as fertilization success. And so sexual selection can drive variation in traits that are associated with morphology, behavior, and even chemical barriers to reproduction. And so once these traits have arisen in populations, they can then facilitate and act as barriers to gene flow. So if you think about a sexually reproducing organism, you can have barriers that can come up anywhere down this line. So it is useful to think of reproductive isolation as a continuum. either pre-zygotic, before you find a mate or the actual process of mating, and post-zygotic, maybe to do with development of a zygote or the survival of the offspring and the eventual fertility of the offspring as well. And so those are some of the questions that interest us at the Reproductive Evolution Lab. And we are based in Singapore, which, if you don't know, it's just off the equator. It's a tropical island and it's a biodiversity hotspot. And so we are interested to ask, what are the mechanisms, especially to do with reproduction, that could be mediating speciation? And so in our lab, we use insects as our model system, and we work on particularly widespread species, and we use both experimental methods, fieldwork, and next generation sequencing technologies to take an integrative approach to look at both the macro-evolutionary patterns of diversification, as well as the micro-evolutionary processes that may be mediating these differences. So before I go any further, the real stars of much of the work I'm going to be showing are the people of the lab, the students. We are very proud to say much of the work is student-led research. And we have students who are working on organisms that they find cute. And so I kind of have to learn what they find cute. So I always have imposter syndrome when I'm talking about insects, because my students often know a lot more than I do. So for today's talk, I'm just going to highlight some of the, a few case studies whereby we've been asking these questions about reproductive influences, reproductive mechanisms that drive variation. So one of the first case studies is going to be looking at sepsid flies. So sepsids belong to a relatively small family of flies. They are found worldwide. And over the last 2 decades, especially, and I'd like to think in large part thanks to the work that I've been doing as an undergraduate and during my master's, is that we've had, with the use of a strong phylogeny, published few comparative studies that show that sexual traits and even mating behavior evolves rapidly in this lineage of insects. And so you can see even here is a male mounted on a female, and instead of just, getting it done, he's courting her vigorously with this come hither movement with his mid legs. And then there are other species that take a more chill approach, just cycling their hind legs slowly during copulation. So behavior can be quite different across this lineage. And so during my PhD, I decided to focus not so much on across species, but more across populations. And so the species that I started to work on then was Sepsis punctum, which is actually quite widespread. It can be found in a lot, even in North America, in Europe, and even parts of Africa and Asia. And all the populations that we work on have taxonomically keyed out as the same species, because they have these diagnostic forelimb armaments that are very species specific, and the genitalia morphology is also the same. But what is interesting is that during my PhD, I recorded this interesting reversal of sexual size dimorphism. So sexual size dimorphism refers to the differences in body size between males and females in a given population or species. And in this species, we found that populations in North America, females were larger than males, which is the standard in most insects. But in populations in Europe, males were larger than females. So since then, we have followed up with quite a number of studies where we've looked at the differences in their development time, what are the proximate mechanisms giving rise to differences in body size, could there be plasticity, how they respond to temperature, are there morphometric differences in this foreleg structure? And so those are the work that I've mentioned here, so you can feel free to check them out whenever you want. But one of the questions that we wanted to investigate in greater detail is looking at the female side of things. What about female remating or polyandry? So polyandry refers to the number of times females mate with multiple males, and the degree to which females remate can directly influence the strength of selection acting on males in that population or species. So this is the work done by our undergraduate Ipeng. So from here onwards, much of the results I'm going to be showing are not published yet. They are undergraduate work that's fresh off the press. So not press yet, fresh off the lab. So this Yipeng asked questions about, do females in different populations mate to different extents? And so she followed focal females, about 20, 30 per population, and she gave them an opportunity to remate with a new male every week for about 6 to 8 weeks. Now that's actually a lot of mating experiments. And then she also tracked their offspring production too. So all I'm going to show today is her raw results on whether they differed in the remating frequencies, and it's quite significant. European populations, females remate a lot more than the populations in North America. She also found this interesting marginal difference within North American populations, whereby the West Coast populations seem to mate a little bit more than the Eastern or Central populations. Now, I just want you to keep those West Coast populations and that color differentiation in mind. I'll come back to it later. So now this gives us a unique opportunity to test and predict, test predictions about population diversification in reproductive traits. For instance, in populations where females rarely remate, then they should males should invest more in mate acquisition rather than post-copulatory sexual selection. And in populations where females remate a lot, you should expect an equal investment in both mate acquisition as well as fertilization success. And so in our preliminary work or work before this, we actually found that is true. In North America, males actually have a unique abdominal courtship that is completely absent in European populations. And what is interesting is that even within North America, we also find a west to east gradient in the intensity to which these populations or the males in these populations are courting the females, right? So once again, that west and east differentiation within North America. So European populations, no courtship, but they are larger in size. So the males are much better at defending territory. So their mating system is slightly different. So instead of courting, they are fighting with each other to defend dung pads because these guys are dung flies. So that's their arena for competition. But what happens after mating, right? So this is a question that Jiawei and Ibo both decided to tackle. So they did numerous common garden experiments as well as condition-dependent experiments where they varied the temperatures, they varied access to resources to try and partition out genetic variation in post-copulatory traits. But for the purpose of today's talk, all I'm going to be showing you is the results from their common garden, which immediately shows that in North America and Europe, there's a big difference in investment in testes. And there's also a really strong positive allometry in Europe. So positive allometry with values larger than 1 means that for every unit increase in body size, European populations are disproportionately investing more in testes than their North American counterparts. And it's not just the testes that are bigger, the sperm is also longer, right? So this is an example of a North American population from New York, and the images are taken at the same magnification, and immediately you can see there's a difference in the tail length. And Jiawei and Ibo also find that there is a significant difference in the trait values. So European populations are producing longer sperm, but the allometric relationships are less than 1. So it's not disproportionately longer, it's just longer proportional to body size as well. And once again, she found, Jiawei especially found this marginal difference in the sperm length within North America, once again, between the Eastern populations and the Western populations. So these populations clearly differ with respect to their investment in pre-copulatory and post-copulatory traits, which is in line with the prediction we had about the differences in polyandry. But what can we make of all this variation? What consequences can that have for gene flow? So as I mentioned, all these flies actually have the same diagnostic traits. And based on CO1 barcoding, they all fall within the same species threshold. And I have to give a shout out to our RA Nicole, who not just the species, the populations that I've shown here, she's actually extracted DNA and barcoded more than 600 individuals across the entire European and North American samples that we have had. And her simplified haplotype map does show some kind of structure in the genetic differentiation. So this is just based on one loci, one gene, CO1 barcode, where we see, can I check if you guys can see the cursor? If I move this? No? So let me move my hands instead. Down, right, that's the bluish colors. Those are the European populations, and the reddish orangish colors, those are the North American populations. So the haplotype map shows some kind of geographical pattern, but it also shows a lot of admixture. Once again, this is just a 300 base pair mitochondrial fragment. It's not going to tell you much about the genome-wide differentiation. So let's look at the genome-wide differentiation. So this is work together with our research fellow, Nalini, who looked at SNPs across the genome for the same populations that I've mentioned at the site. And what we find is that there's clear differentiation, not only between the continents, which is shown on PC1. So here PC1 is on the y-axis, and that differentiation separates out North America and Europe. And then PC2 separates out the Eastern populations from those Western populations that were so different with respect to their reproductive traits. And what is interesting is even within Europe, down here, we see a separation between that purple group, which are Italian populations that are south of the Alps. So clearly geography is also playing a role. So it is likely that that the case that is going on in sepsis punctum is a mixture of both geographical variation, reproductive differentiation, and incipient speciation. So that's pretty much the take-home message. Right? When we take an integrative look, looking at behavioral data, morphological data, and genomic data, what we can see is that there is limited gene flow across these populations, and that the differences in mating system could also play a role in mediating incipient speciation. So what's next? Right, so the insect reproductive tissues, especially the male insect reproductive tissues like the testes and the accessory glands, are often known as hotspots for novel gene evolution, for rapid evolution too. So these accessory glands are responsible for seminal fluid proteins that can influence female behavior or female fecundity even. And then of course the testes are responsible for sperm production. So our postdoc is currently in the midst of looking at differential gene expression, as well as the birth of novel genes or genes that as far don't have any known function yet. So this is still work that is in progress, but the exciting thing is that we have managed to do this for a species that does not have a reference genome. So she has kind of developed novel de novo pipelines that can be used to look at novel gene evolution in non-model systems. And what is also interesting is that male bias size dimorphism is a derived trait in sepsis. In most species, females are the larger ones. And we have in this clade an independent reversal in another species known as sepsis neosynapsia, further up there. So the star refers to punctum, which is what I've been talking about. And in neosynapsia, it's reversed in the other way. So in punctum, males were larger in Europe. In neosynapsia, males are larger in North America. So we have this interesting independent example where we can, whatever we did for punctum, we can try to do that again for neosynapsia and see if it fits our predictions about differences in mating behavior and barriers to gene flow. So that was a case study where there's a species that has had so much taxonomic information. I knew for a fact that these are the same species, but I had all these checklists and all these taxonomic keys that I could follow. But how are we going to study ecological and reproductive mechanisms underlying speciation when your species are not described? Right? So this is an example of a micropisit from Singapore, which you find all over the place, but we don't have a species name to go with it. And in fact, several studies suggest that, there's only about 20% of known all insects that have been described so far, and that's about 1 million. So you can imagine how many more species there need to be described. And the situation is particularly dire for the tropics, because even in this recent review paper in 2019 that looked at about 73 studies looking at insect declines, only 3 were from the tropics. And that's because we don't really have that many comprehensive surveys in the region. And even within the tropics, a lot of the fauna is based off primary rainforests. So a lot of the areas that are mangroves, for instance, or secondary reforest, or areas that have been rewilded, all these areas have lesser attention that is paid to them. So as someone working in Singapore, looking at insect biodiversity, there are several challenges. The lack of checklists, we don't have updated keys, and the keys that we have are from regions like Thailand, which are quite different from our geography and our climate slightly. We have unresolved taxonomic issues. We have morphologically cryptic or similar females, and also cryptic species complexes. So for instance, let's say I'm interested in dung beetles, right? I'll put out a trap with a baited pitfall trap here with some poop, and I get I get a lot of beetles. So these beetles can then be sorted to morpho groups with, of course, consultants from, who have expertise in the region. For instance, Eleanor Slade at NTU, who's has done so much work in Sabah, for instance. Tariq Azigo at the University of Malaya, working in Peninsular Malaya. And of course, Darren Mann with the Oxford Museum, who has had a lot of samples from across the span, across Southeast Asia. But even after consulting with a lot of them, there is still a lot of questions that are unresolved. There's only a few species that we can, without a doubt, say that this is that species. Most of the time, we say it looks like this, or it could be this species. And in many cases, they don't even have a species name. So what can we do? Oh, of course, this is also just mainly based on males, because the females are quite similar looking. But we can take an integrative approach which complements our morphological study. So we use DNA barcoding, looking at the cytochrome oxidase subunit 1 fragment. And by using this barcode, we can actually then match male and female specimens, and we can resolve cryptic species complexes. And we've used DNA barcoding as a tool to help us in our ecological studies, as well as even co-evolutionary studies, looking at host-parasite interactions in not just dung beetles, but in other insect groups in Singapore too. So how does that work? Well, basically you take a part of that insect that you're interested in, you squish it up in some lysis buffer, and we use next generation sequencing methods. So we do them in plates. We have unique primers for each sample. And so you pool all together and you run it in a single lane. And then bioinformatically, you can filter out the reads of interest. And then you can cluster them based on intraspecific and interspecific genetic differences, right? So based off whether individuals that fall within a threshold, let's say less than 3%, they are likely to be the same species. Individuals that have larger differentiation, likely to be different species. And of course, with this method, we can not only double, but we in fact tripled our number of specimens that we could actually identify as operational taxonomic units. And what we find is that we ended up being splitters. I didn't think we were, but we overestimated sexually dimorphic lineages and underestimated monomorphic or cryptic species. And so when you have cryptic species, then that's something interesting, because you can get cryptic species either because of convergence, meaning the environment is similar, so I independently evolve the same morphology, or you can have really rapid speciation, whereby the genomic differences are accumulating much faster than morphological differences. And so that's the second case study I wanted to tell you guys about, looking at Catasius Malosis and looking at potential reproductive trait diversification in this cryptic species complex. So before I do that, let me share a little video that features our students. Now, the video is going to be in Mandarin, but there are English subtitles for you to follow along.
屎壳郎一般呢寿命为3年，最小的品种的体长只有3毫米。它们除了忙于维持自然界里的清洁卫生，在农业里呢其实也扮演了重要的角色。根据美国生物科学研究所的报告，屎壳郎为当地的养牛业每年节省了3亿8千万美元的粪便清理费用。
这里是哪里？到处是一片白，不会是天堂吧？
不是啦，我听说这里是很重要的地方，专给人类研究如何保护我们的。哦。
不在森林里寻找屎壳郎的时候，叶直松大多时间都待在实验室里探索爱虫的秘密。
So this is a setup for breeding live dung beetles. You can see them come to the surface, take the dung and tunnel back under. And sometimes you can see the little tunnels.
不同品种的屎壳郎有不同的习性和需求，所以要通过研究增进对不同品种的了解，才能针对性地进行保育工作。 叶直松和学生们靠着不断努力，至今为止已在本地找到了大约20多个品种的屎壳郎。
So you can see in different species, there are different shapes of their horns. And some of them actually look very, very similar just based on external appearance. But based on DNA, they are actually very distinct.
哎呦，被你发现了！是啊，我们在全世界有超过8000个品种，是最多品种的甲虫之一哦。
Once you learn more about these things, you really see the big picture, how everything is interconnected. And that only adds more to its beauty and not take away from it.
我还有很多秘密等着你们发掘，但现在我要回家喽，下次见。
So that's the beetle that just went back in, and let's look at it a bit more. This is Catasius Malosis. Well, it's one of the large nocturnal tunnelers, and it is actually the largest dung beetle of record in Singapore. Now, for those of you, saw that 20 species of beetles, you're like, oh, that's not a lot. But Singapore is tiny, and records previous to this only thought there were only about 10 species of dung beetles in Singapore. So with our technique, we managed to double the estimates of species in Singapore. And particularly this cryptic in Catasius Malosis, we found a potential cryptic species, meaning species that are different based on their CO1 barcode. So this is, in fact, Sean, the PhD student, our star of the beetle work that we do. He went to the Oxford Museum and he got really excited knowing that actually Wallace himself collected a Catasius specimen on Singapore a long time ago. But so a lot of surveys in India, China, and Southeast Asia often classify all of them as Catasius Malosis as a single widespread species. But of course, in our mitochondrial assessment, we realized that there were actually at least two different clusters. A common cluster, which we found quite often in Singapore, but often in the same trap, you would find beetles that were genetically different from the common cluster. And those we would classify as rare. And what is interesting is that we also have some populations that are very genetically similar. There's no admixture whatsoever. For instance, the Langkawi population, which is an island off the west coast of Peninsular Malaysia, that is almost entirely yellow. That's Langkawi. And the specimens from Sabah that were donated by Eleanor Slade, they are also their own haplotype in purple. So the two extreme ends of that range seem to be different. And so that's actually one of the limitations of CO1 barcoding. Because when you just have a few samples that happen to be at the far ends of a spectrum, you can get them as being completely different species because they are far apart. So with a 5% difference, if you compare Langkawi and Sabah, different species. But then you also have individuals that from Singapore that cluster with Langkawi and that cluster with Sabah. So this is known as a triangle inequality that can arise when you have, let's say, recent speciation events or introgression events, incomplete lineage sorting, or even oversplitting, right? So one way to deal with this is to do what we did earlier. We can look, oh no. Are you guys not able to see the? All right, so take my word for it, they are separate clusters. Oh, technology. So what we end up having is the Langkawi was clustering by itself. We found a yellow cluster, and then we found a purple cluster by itself. So it reflected the mitochondrial clustering. And we found the yellow, the pink and blue were a separate cluster on their own. So it seemed to show that we're almost 3 different clusters of Catasius that we are finding in our region. So then we had to try and ask, could there be any morphological differentiation that can explain this genomic variation? So Sean once again took a large approach looking at not only just body size, but also shape. So he looked at pre-copulatory traits as well, like the horn shape, as well as post-copulatory traits such as genital shape, right? And what he found is that actually there was body size differences in a cline that was differentiating between the Western populations and the Eastern populations. And interestingly, there was significant difference with respect to wing loading. So wing loading is something that can explain dispersal. So individuals that or populations that differ in their wing loading may not be able to disperse as much as individuals that have, let's say, a larger wing space so can capture more wind during dispersal. And so the populations in yellow and purple that are genetically differentiated are also different with respect to wing loading. But interestingly, we didn't find any difference with respect to the reproductive traits. So the trait allometry. So if you look at just the raw values, head shape or genital shape by itself, they do differ. But if you take into account allometry, meaning since body size is different, let's account for allometry. And when you account for allometry, there's no significant difference between the different populations. So where do we go after this? Well, one of the reasons we couldn't do widespread sampling was, of course, due to COVID restrictions. And so I have to give a shout out to Sean, who managed to still hang in there and pull through despite an entire chunk of his PhD not being able to come to fruition. So we would like to follow through with wider sampling across the range and potentially look at ejaculate traits. Because in a recent assessment, when we got live specimens, we realized that their sperm tends to conjugate, and there were differences in the sperm head morphology between some of the populations that were in the common cluster versus the rare. And in fact, our postdoc, Nalini, also looked at gene expression and found rapid genomic evolution in these 2 male structures, the seminal vesicles and the testes, which are responsible for sperm, and as well as the accessory glands, which are responsible for ejaculate traits such as seminal fluids. So with that, let me take a slight shift. So all this while, we've been talking about ecosystem service providers, but there's also insect reproduction that is relevant to looking at potential solutions that can be sustainable for human problems too. For instance, disease transmission. So I'm in a tropical area that has lots of mosquito-borne diseases, and diseases like dengue and chikungunya are endemic to my region, right? And despite long-term efforts at vector control, we still have cases that are persisting in Singapore. And so much of the research has been focused on the molecular mechanisms of actual disease, infection, but not a lot has been done about the vectors themselves. So in Singapore right now, the government and the National Environment Agency have embarked on a biocontrol that takes reproduction into account, specifically using Wolbachia. So Wolbachia is an intracellular reproductive bacteria that can influence the ability of wild-caught females to lay fertile eggs. So in Aedes aegypti, which is the primary vector of dengue in Singapore, they naturally don't have any Wolbachia. Wild populations don't have them. So if you take males, you inject them with Wolbachia, you release them, and these males mate with the wild females, their eggs are inviable, or they are not even able to lay eggs sometimes. So this image down there is actually our Prime Minister on his Instagram, putting his hand into a cage of male mosquitoes just to convince people that releasing male mosquitoes is fine because they're not going to bite you. But they released the females too by accident. But what is the problem? The problem is Aedes aegypti is not the only species that is likely causing dengue in Singapore. We also have Aedes albopictus, which is actually native to the region. And Aedes aegypti, which is invasive, tends to hang out in the urban areas. Albopictus can actually take advantage of any kind of habitat. It is adaptable to land use change. It can be found in forests, in managed parks, and even in urban environments. And one of the concerns now is that with the suppression of Aedes aegypti, are we going to have Aedes albopictus taking over these niches? And will Wolbachia even work for Aedes albopictus? So, for instance, one of the things you have to ask is, if a female is mating with the Wolbachia male, and she remates with a non-Wolbachia male, can that still rescue her fitness? So the first question is, do females remate? And much of the previous research from other parts of the world seem to suggest that mosquitoes don't really remate. But we didn't quite believe that, because we could actually find various biased paternity differences when we looked at microsatellite of offspring. So Hillary, our master's student, decided to go out and collect wild females and look at their sperm storage organs. Right? She looked at in the coastal wetlands, in the nature reserves, in the peri-urban as well as urban environments, because albopictus can be found everywhere. Right? And so she dissected out the female reproductive tract. She took a look at the morphological variation as well. And she took the sperm storage organs, these spermatici. Then she crushed them in some lysis buffer. Using about 9 polymorphic microsatellites, she tried to ascertain how many different males this female has mated with. So she also looked at differences in male traits, but I'm not going to focus so much on that for today's talk. But just to say, the sperm is kind of cute. So what did she find? Well, first and foremost, she found remating estimates that are much higher than what's been reported in the literature. The highest report so far was about 26% in a population. She found about 80% of her females had sperm from more than one male. And in some cases, females had sperm up to 5 different types of males. Right? So these actually, then our question is, could remating behavior influence gene flow like the way we saw in Sepsis punctum? We can't ask that question with just 9 microsatellites. So then Huizing, our graduate student, comes in and takes this to another level, right? She sampled across the island at a finer scale, looking at forested areas, managed parklands, urban areas, and she did what we've mentioned before as well, looking at genome-wide data, right? So she finds actually across the genome, even in a small island like Singapore, we're only 700 square kilometers wide. It's tiny, tiny, tiny. And yet, even in that tiny island, we can find fine-scale population genomic structure across different habitats. And what is cool is that you can then map this onto the landscape differences of Singapore. Because Singapore has a lot of fragmented forests. So these forests end up becoming barriers to gene flow between urban estates that are on either side of the forest. So these on that map of Singapore, you see those red areas, those are forest patches, and those are barriers to gene flow. And the gray areas are corridors to gene flow. So urban estates and public housing estates that where populations are admixing all the time. And what is interesting she also finds that there is a correlation between their natural Wolbachia levels in these species. So unlike Aedes aegypti that did not have any Wolbachia, Albopictus not only has Wolbachia, it has two different strains of Wolbachia. And the different strains and the loads vary with the populations that are found in different habitats. So if we were going to use the same Wolbachia strategy for Albopictus, it's probably not going to work. So what's the takeaway here? That heterogeneous landscapes and potentially even reproductive microbes may be playing a role in dispersal and gene flow in even in a small geographic area. And so where are we going after this? Well, this is actually what, oh, my student's face is missing. There's supposed to be a student's face there called Tyrone. Tyrone is a new grad student who will be joining us, and he's going to be looking at what do the populations differ in terms of their viral loads or their vector capacity? Is there potential competition with other mosquitoes? Are there any ecological factors that can underlie these corridors and barriers to gene flow? Because what we find is clear structure in communities of different mosquitoes. Like in urban areas, you find some assemblages, in the forest areas, you find others. So that's kind of the work he's going to be looking at down the line. And finally, we've also taken insect reproduction to look at waste management and sustainable food production. So in Singapore, we have a huge food waste problem. And one of the recent advances or recent push by the government is to try to be more sustainable with our food production too. Right now, 90% of our food comes from elsewhere. So by 2030, the government wants to produce at least 30% of our nutritional needs. But where are you going to get this if you're not an agricultural country? Well, you can transform, public housing to have facades that have vertical farming. You can have ponds that can use freshwater fish rearing. So this is the project that we embarked on.
Singapore is a food paradise, but it also has a messy problem with food waste. In 2020, Singapore generated more than 650,000 tons of it. A lot of food gone to waste for a city of just 5.8 million people.
Part of our research that we're trying to do here is try to reduce our negative impacts on the environment. So that's the big picture. And why not use these humble insects who've already been doing this for millions of years? They've been converting waste very efficiently. Now, why not modify them for our purpose so that they can help us in, well, reducing our own environmental footprints?
Nalini Puniamoorthy cultivates a special breed of black soldier flies. To thrive in a land-scarce city like Singapore, the flies must learn to mate in much smaller spaces.
So black soldier flies generally mate in really large spaces, about 10 times the size of this cage here. Now, through selective breeding, we've managed to reduce the amount of space that's required to establish a culture of black soldier flies. They then mate the next generations. So what we have here is little substrates that mimic food waste. So they attract the females to come there. And so when the females are attracted, then the males then have a night out. It's by trial and error. Most of science is by trial and error. And that's how we've managed to come up with Singaporean lineages of flies that can mate in much smaller spaces.
So I don't have time to feature the ongoing research that our other grad students are working on. For instance, looking at the plasticity of the reproductive traits when you rear them on food that is as diverse as food that is in Singapore. We have very heterogeneous food because we have so many different cultures, and our food waste is also different in terms of spice and salt level. We use a lot of soy sauce, as well as difference in the nutritional composition too. And so Shakti, another grad student, is going to be looking at the microbiome, especially the reproductive microbiome, how that varies in response to environmental challenges. And Adrian, another grad student, will be looking at potentially using AI to kind of predict what are the variables that can influence black soldier fly rearing for an urban environment. So with all of that, what's the takeaway? Insect sex is kind of cool. And you can actually understand barriers to gene flow by looking at both the environmental components as well as the reproductive mechanisms. Integrating behavioral, morphological, and molecular data can be a really powerful tool. And the real warriors are the students. If we can nurture and give students agency in their research, where they are taking the steering, right? They are leading these projects, then you can have a really nice research program where everyone's feeding off each other, even though they work on completely different organisms. And so that's kind of what we try to do at the Repro Lab in NUS. If you want to know more, you can go and check out our website. And once again, I just wanted to end off with the real warriors or the heroes. These are the students who are doing the work. They are out in the field. They are doing all these crazy morphological measurements and behavioral studies. All I do is say, why don't you do that? And they do it. So I'm very fortunate to have a really good group that I work with. And thank you for your attention, and thanks to people who funded us.
Okay, thank you very much, Nalini. I'm going to open it up to questions. I don't think it's on.
That's better. Hi, Liz Duncan, University of Leeds. Thank you very much for a beautiful talk. Insect reproduction, I think, is one of the most fascinating things. I have a question about the first system that you presented. I was really intrigued with your DD-RAD-seq data, and I wondered whether you thought that data was able to provide enough information. Basically, can you get any clues about how the morphology has changed from that DD-RAD-seq data?
Yeah, so that RAD-seq, the very first example, the biggest difference in the morphology was the size dimorphism, right? And of course, the reproductive traits that can be accounted for in some ways due to the larger size. So the RAD-seq data, all it showed was that Europe and North America were different. So that can explain the size dimorphism. But within North America, there's also a differentiation, but they have the same size dimorphism. They do differ, however, in behavior, and they do differ in the degree to which females remate and the degree to which the male sperm may be marginally different, might be different. So the RAD-seq data tells us across the geographical cline, yes, there's differentiation, but it's not necessarily pointing out within continental variation.
And I'm guessing because you don't have a reference genome, you can't link it to particular genes or anything at this stage.
No.
Maybe in the future.
Yes. Thanks.
Oh,
sorry. There's also questions.
Hi, I'm Will Nash from the Institute, and thank you for an awesome talk and so much work presented. I was also wanted to ask about the sepsis system. Super cool. Do the European and American populations still hybridize?
Yes, I was waiting for this question. So in the lab, you can get them to mate, and the F1s do perfectly fine. In fact, as I said, the European populations don't have a abdominal courtship. The North American ones have. So the F1s do like a half shake. It's like a half Shakira move, right? So the F1s do show it's probably additive genetic variation for that trait. But what is interesting is that at the F2, we may be getting some leakiness. They do produce F2, but we have not been able to get F3s and F4s. So part of the work would be to try and get a genome, so we could do potential QTL studies and find the underlying loci that might be responsible for that. Thank you.
Whilst Electra is running between people wanting to ask questions in here, we'll just go to one of the questions online from Christopher Williams. Is there sperm competition in sepsids?
Well, we think there is, because females mate multiple times, and so there is sperm that is stored within the female reproductive tract. What is cool is that females actually have two types of sperm storage organs, right? They have one for long-term storage, a lot like what we heard from Jessica, right? But they also have a short-term sperm storage organ, and that organ actually seems to evolve very fast across the entire sepsid lineage. So it could be acting as a selective mechanism to bias paternity. And what we find is that it's not really clear if it's raffle or there is displacement from last male sperm precedents. So we think there is sperm competition, but we have not been able to disentangle what's mediating. Is it female or is it male only effect? Did that help answer your question, Christopher? Oh, I realize he can't respond.
Okay. We'll take another question over here.
Yeah, Rosamund from Lancaster University, and I'm going to move to the dung beetles system because I work on dung beetles. So I'm interested to know if you have done any studies with your species about nesting behavior, if that has changed. So if they change, maybe the barrier is related with nesting behavior and pairs helping each other.
I completely agree. I think there's a lot we don't know about the ecological variables that are driving, potentially driving, because these guys are nocturnal, so finding them is also tricky, but they also tunnel meters down. So trying to have these microcosm experiments out in the field is hard. One thing that we are hoping to do is collaborate with Eleanor Slade in NTU, and she has microcosm experiments planned in Sabah. And so Sabah has these different phenotypes. So maybe we could potentially test it out on her system because they are within the same complex. Right? So we try it out there, and if it works there, maybe we can try it here.
That's a great idea.
Thanks.
Are there any more questions from here? I have one.
Yes.
So going back to the sepsis flies, I was intrigued that there's appears to be, from your photographs, quite a big difference in sperm length between the European and the American populations. Is there any evidence of variation in female reproductive architecture?
Oh, I love that question.
between the two populations or the two continents?
Well, that's what I had hoped for. I had hoped for a potential correlation between a female reproductive trait and the male reproductive trait that is co-evolving with it. So maybe females are exerting a selective pressure, but we didn't find anything that wasn't just allometric. So if it was, let's say, disproportionately longer than the North American populations, then it's not just an allometric relationship, that's hyperallometry. But here, the difference in female structures can be completely explained just by body size. Yeah.
Okay. Thank you. Are there any other questions? Okay, I think a round of applause. Thank you.
Thanks, everybody.
Thank you. That was great. That's brilliant.